Bismillahirrahmanirrahim. Assalamu alaikum and welcome to Biosciences with Shatreen Sawati. And Shatreen Sawati is here to deliver the lecture from my coronavirus series. And this lecture is about the COVID-19 variant. And you all are so much curious to know about the COVID-19 variant. And all the students and the researchers want to know which mutations have been detected in the SARS coronavirus 2 to be the B117 or the UK variant. I will discuss in my this lecture about the COVID-19 UK variant which is B117, and in my next lecture I will discuss about the P12 which is Brazilian variant in detail. I will discuss about the genetics of all the variants which is going on and which is detected by the which are detected by the scientists throughout the world. So this lecture is the important piece of the research which I created on the basis of the research which is going on throughout the world. So I'm thankful to all the researchers for providing me such an information, and I created this lecture. So in this lecture I will discuss about the B117, which is the UK variant and very dangerous and more you can say the most easily transmittable, and many more I will show you. I will discuss the genetics of the SARS coronavirus 2, then B117 lineage, and which sorts of new mutations are going on. So first of all, I will discuss about the genetics. As I have discussed about the genome of the coronavirus in detail in my previous lectures and about the structure of the coronavirus, so must watch all these lectures, then you will be easily understand this lecture about the mutations and about the coronavirus variant. So as you can see here, this is the structure of the SARS coronavirus 2, and it consists of the outermost spike proteins, spikes, which are very important, playing role in infection and attachment to the host. And inside the envelopes you can see here the single-stranded RNA, which is the positive sense, and I will not discuss in detail here because I have discussed in my previous so much. Must watch my lecture about the SARS coronavirus structure and genome and spike protein and many more. So genetics. According to the genetics, mutations in the SARS coronavirus are common. Over 4,000 mutations have been detected in the spike proteins alone. Must keep it in your mind. 4,000 mutations have been detected. And if you people need all the research articles, I will provide you the links in the description box. You can easily download all these research articles on the basis of which I created this lecture. So how much? Over 4,000 mutations have been detected in its spike proteins, and according to the COVID-19 genomic UK. So the B117, which is the UK variant, is defined by 23 mutations. 23 mutations have been detected in B117. 14 mutations are non-synonymous, 3 deletions, and 6 synonymous. And if you are not familiar with the synonymous and non-synonymous, I will give you some information about the synonymous and non-synonymous and the differences in this lecture later on. Okay. So this is the structure, and as well as you can see here the genome of the coronavirus, which is single-stranded RNA, and I have discussed in detail the single-stranded RNA, which is the genome of the coronavirus, consists of 6 open reading frame, and 2 open reading frame, A1A and 1B, they are responsible for for non-structural proteins, and from spike proteins S, E, M and N are responsible for the structure proteins. Non-structure proteins means they are synthesized enzymes and proteins and many more. And scientists have detected 23 mutations in its genome, and you can see here I have mentioned all these like the deletions and in the open frame A and in the spike proteins and many more. So all these mutations are responsible for the emerging of the new coronavirus, which is B.11.7, which is also known as UK variant, which I'm going to discuss here in this lecture. So here I am sharing Elizabeth Pharmacon Lab announcement with you that the Elizabeth Pharmacon Lab has discovered a new strain of the coronavirus. Czech biologist Omar Seri, who is the company founder, said on Twitter. Thanks to the sequencing of the samples from the patients with SARS coronavirus 2 infection, our laboratory discovered hitherto undescribed mutations of the spike proteins. So this is the statement given by the Omar Sari. Okay. The epidemiologist explained that the new mutation does not appear to be more contagious and COVID-19 vaccines should be or may be effective against it. So, fortunately, the mutation is in a position, this is the statement from the Elizabeth Pharmacon lab, that fortunately, the mutation is in a position that should not affect the binding of the antibodies or cellular receptors, Sari said on Twitter. Is it clear? So this is the announcement of the Elizabeth Pharmacon lab. I just want to share because they shared on the Twitter. So I am sharing with you. B.11.7 lineage. Now I am going to discuss in detail about the B.11.7. This variant has a mutation in the receptor binding domain. Now, if you are not familiar with this word receptor binding domain, so must watch my lecture about the spike protein, and I have given complete information about the receptor binding domain. So the variant has a mutation in the receptor binding domain of the spike protein at position 501. Now 501, which is what is 501, I will show you. This variant also has served other mutations including 60, 70 deletions occurred spontaneously many times and likely lead to a conformation changes in the spike protein. And P68 1H mutation near the S1S2 furin cleavage site. Now, what is furin cleavage site? I will discuss later on. Furin cleavage site. Furin is an enzyme which is responsible for the cleavage of the spike protein, a site with the high variability in coronavirus. Is it clear? This mutation has also emerged spontaneously multiple times. Okay. Mutations in the RBD can change antibody recognitions and ACE2 binding specificity and lead to the virus becoming more infectious. Is it clear? So the changes in the RBD domain change the antibody recognition as well as ACE2. So maybe the vaccine will not be effective. I will share some vaccines with their efficacy against the B.11.7 later on. Okay. This variant is estimated to have first emerged in the UK during September 2020. Since December 2020, several countries have reported cases of the B.11.7 lineage, including the United States. This variant is associated with increased transmissibility, that is more efficient and rapid transmission. In January 2021, scientists from UK reported evidence that suggests the B117 variant may be associated with an increased risk of death compared with the other coronavirus variant. Early reports found no evidence to suggest that the variant has any impact on the severity of the disease or vaccine efficacy. Okay. Now you can see here the transmission electron micrograph of the B117 variant coronavirus. The variant increased transmissibility is believed to be due to the changes in the structure of the spike proteins shown here in the green. You can see here these are the spike proteins which with more mutations, and these mutations are responsible for its transmissibility as compared to the other variant of the coronavirus. Okay, mutations. Now I'm going to discuss, very important. Must watch carefully and note down all these mutations in the SARS coronavirus. I will give you information about the synonymous and the non-synonymous mutations here. So the types of mutations are non-synonymous mutation are the, you can say, the non-synonymous substitution is nucleotide mutations that alter the amino acid sequence of the protein. Must keep it in your mind. The non-synonymous substitution is the nucleotide mutation, and the synonymous substitution, often called a silent substitution or the mutation, though they are not always silent, is the evolutionary substitution of one base for another in an exon of the gene coding for a protein, such that the produced amino acid sequence is not modified. In this case, not modified. But in the non-synonymous substitution, it is modified. I will show you here. Now you can see here, this is the synonymous mutation and synonymous substitution, you can say. So in synonymous substitution, you can see here, adenine base is changed with this guanine. So just one. But in this case, the complete nucleotide is changed. So if the complete nucleotide change here, the amino acid sequence change. But in this case, no change in the amino acid sequence. Is it clear? And sometime if the mutation occur in this way, like the UGA, which is the stop codon, it will stop the formation, or you can say that the coding for the amino acid. So this is the non-synonymous and synonymous substitution. In genetics, there is a missense mutation. What is missense mutation? Missense mutation is a point mutation in which a single nucleotide change result in a codon that codes for a different amino acid. It is the type of the non-synonymous substitution. Is it clear? Okay, so this is just information for the non-synonymous and synonymous and missense mutations. Okay, now I'm going to discuss the first mutation which is detected by the scientists, by the researchers in the new variant B117. This variant has a mutation in the receptor binding domain of the spike protein at position 501, where the amino acid asparagine has been replaced with the triosine. Must keep it in your mind. At the 501 position, what will happen? Asparagine has been replaced by the triosine. There is a replacement. You can see that the shorthand for this mutation is N501Y. So it is written, or you can say the abbreviated as N501Y. Why it is called 501? Because the mutation occur at this position, at which the asparagine has been replaced with the tyrosine. Asparagine is replaced by the tyrosine. Must keep it in your mind. So the next E484K mutation. So the mutation occur at 484 position. This mutation also found in the SARS coronavirus 2. So the name of the mutation E484K refers to an exchange whereby the glutamic acid, like you can see now the comparison in N501Y, there's a replacement of the asparagine with the tyrosine. Asparagine with the tyrosine, and now here the glutamic acid is replaced by the lysine at 484 position. So it is nicknamed as EEC. E484K has been reported to be an escape mutation, that is a mutation that improves a virus ability to evade the host immune system from at least one form of the monoclonal antibodies against SARS coronavirus 2, indicating there may be a possible change in the antigenicity. Is it clear? Now, first you learnt about the N501Y, and now about the E484K mutation, and now P681H. In January 2021, scientists reported in a patient that the mutation P681H, a characteristic feature of the significant novel SARS coronavirus 2 variant detected in the UK B117 and Nigerian B11207, is showing a significant exponential increase in worldwide frequency similar to the new global prevalent D614G. This is also the type of the mutation. So the P681 or P681H is a proline to histidine substitution at amino acid position 681. Now the concept is clear upon you people that why it is called P681H mutation, why it is called E484K mutation, why it is called N501Y mutation. Now I hope so the concept will be clear upon you people. So what is the D614G? D614G is a missense mutations that affect the spike protein of the SARS coronavirus 2. The frequency of this mutation in the viral population has increased during the pandemic. In this case, glycine has been replaced by the aspartic acid at position 614 in many countries. Is it clear? I hope so the concept will be clear upon you people. Now the 5 things you need to know about the B117, the new coronavirus strain. Data suggests that B117 is about 50% more contagious than the original strain. The risk for complication to the infection is similar to the original strain. It is probably more widespread than we realize. Both COVID-19 vaccines that have been received emergency use, and I have delivered a lecture about this, about the Moderna and the Pfizer's, which is messenger RNA vaccine. This vaccine has been received authorization by the US Food and Drug Administration for effective at providing the protection against the original strains of the coronavirus. Results of more recent studies suggest that they will also provide protection against the B11.7. The vaccines are scientifically proven to be safe and effective. Keep practicing COVID-19 safety measures like wearing mask and washing hands, and keep the distance of about 6 feet and many more. Okay. Thank you very much. I hope so the lecture will be clear upon you people. If you have any question, write in the comment box. I will try my best to give you the answer. So thank you very much and Allah Hafiz.